Well, I'd like to say just a little bit more about what happens as information is passed from the nucleus of the solitary tract through the thalamus, and then from thalamus up to the cerebral cortex. So when information is processed in our taste regions of the insula, it's integrated with visceral sensory signals that are also processed in this insular cortex. And that information then is passed on to what we might consider to be higher order associational areas, especially in the orbital and medial parts of the prefrontal cortex. And I would highlight one very interesting region of the orbital cortex of the brain. So here's the orbital surface. It's that part of the brain that overlies the orbits of the eyes. Here again is our olfactory bulb and our lateral olfactory tract, just for a little bit of reference. I'd like to draw your attention to this posterior part of the orbital cortex. This, we think, very well may be the first place in the cerebral cortex where flavors are represented. This is a site anatomically that's receiving input from our gustatory pathway, as illustrated in the figure before us. And if you'll recall our discussions about the olfactory pathways, it's also a region that receives input from our primary olfactory cortex, such as our piriform cortex. This is also an area that is going to be integrating signals derived from our trigeminal chemosensory pathways, as well as other higher order information from the somatic sensations associated with feeding. So when we put all that together, what we have is flavor and a sense of the pleasure derived from that food, be it of positive or negative valence. So we talk about the hedonic value of food as being extremely important in reinforcing feeding behaviors, and this perspective on the value of food seems to be represented in this part of the brain, this posterior part of the orbital cortex. Well, information from the cerebral cortex is also passed on into structures like the amygdala, as well as the hypothalamus, and so we can appreciate from this anatomy, I think, how information about the foods we ingest can be distributed to a broad cortical network that's involved in representing the signals derived from inside of our bodies and integrating that with our conscious evaluations and judgments about the significance of these stimuli. Well, we'll talk much more about these topics when we get into the final unit of the course, when we talk about cognition, the associational cortex, and some of the truly fascinating issues in modern neuroscience that revolve around our sense of judgment, our sense of value, our sense of self, and how much of this is derived not just from what we see and what we hear and what we touch in the world that's beyond our fingertips, but also the world within. These sensations, including the chemical sensory signals and our visceral sensory signals that become integrated into our conscious sense of self. So I hope you'll enjoy that discussion. Stay tuned for that. Okay, now I think we're ready to turn our attention to the problem of sensory transduction in the gustatory system. So sensory transduction again refers to the challenge of converting mechanical energy, electromagnetic energy, or molecular energy into electrical signals within a nerve cell. So we've seen how that happens in each of the sensory systems, and let's consider now how it occurs in the gustatory system. So I'll just remind you that the tastants, that is the actual molecules that will activate this system, obviously are ingested, taken into the oral pharynx, where they can interact with specializations of the epithelium. We actually find these taste buds in specializations that we call papillae. So at the back of the tongue, we have a row of circumvallate papillae. At the sides of the posterior tongue, we have a set of foliate papillae. And then in the anterior part of the tongue, we have what's called fungiform papillae. And these names are just anatomical terms that describe a bit of the morphology of these papillae. So don't really be concerned with the precise configuration of this tissue that forms these papillae, unless this is of particular interest to you. But otherwise, I just want you to know that there are these papillae that are distributed in different ways across the surfaces of the tongue and then in the posterior part of the oral pharynx. Now these posterior regions in the esophagus and in the soft palate are going to be innervated probably by branches of the vagus nerve, nerve 10. Here on the tongue, the posterior third of the tongue is going to be innervated by branches of nerve 9, the glossopharyngeal nerve, and the anterior 2/3 or so of the tongue, including these fungiform papillae, these are going to be supplied by branches of cranial nerve 7. Okay, so this accounts for how nerves 7, 9, and 10 provide gustatory signals into that nucleus of the solitary tract in the brainstem. Well, now let's look at the anatomy, the microanatomy of one of these papillae. So what we see is that there's a bit of a trench, so we can imagine that water-soluble tastants work their way into these trenches where they can interact with these taste buds, these specializations that we find in the lateral flanks of these trenches, and it's within those specializations that we find our taste cells. So here's a closer look at a taste bud. So this is the tissue that we find in the tongue. These taste buds include our sensory receptor cells, our taste cells, and they also include a variety of basal cells and supporting cells. Now the taste cells are the ones that, of course, I would draw your attention to primarily. These are neuroepithelial cells that have an apical surface that comes together into something that we call a taste pore. So it's specifically through this taste pore that our ingested molecules would interact with the apical surface of our taste cells. And it's at that apical surface that interact with the tastant molecules, leading to the depolarization of those taste cells, the release of neurotransmitter at a synapse between the base of the taste cell and the axons of those 3 cranial nerves, nerves 7, 9, and 10. Okay, so now let's focus in on this apical surface of the taste cell and understand how sensory transduction happens. Well, for a taste cell, sensory transduction happens through a variety of different mechanisms. And as I'll tell you in just a moment, the receptors that are expressed help to define the response of that taste cell. And cells that are tuned for particular molecular forms, thereby giving rise to particular gustatory sensations, are distributed in overlapping but somewhat biased regions over the surface of the tongue and in the posterior part of the oral pharynx. Well, I'll tell you that story in just a moment, but first I want us to focus on the interaction between our tastant molecules that are going to be presented to this taste pore, the apical surface of these taste cells. And we know that there are lots of different flavors out there, right? Well, psychometrically, we can categorize them basically into 5 principal flavors. There are tastant molecules that will induce these flavor perceptions, and those 5 basic categories are salt, acid or sour, sweet, bitter, and just in the last couple of decades, I think psychologists around the world have agreed that there's a 5th category that we call umami. And umami refers to the taste of monosodium glutamate and probably other related amino acids. It's derived from a Japanese word that means delicious. This is probably why some of us enjoy a little bit of monosodium glutamate in our diet. I hope you'll appreciate now that too much monosodium glutamate can be a very bad thing. It can induce excitotoxic injury, but in small amounts, it is sufficient to activate this taste pathway that gives rise to this, apparently for most of us, very pleasurable sensation. Okay, so the point here is that there are different categories of tastants that interact with different taste receptors. And these receptors have quite distinct mechanisms for transducing this information. For the salt and the acids or the sour tastants, they interact with ion channels that have receptors on the extracellular face of these receptors. So when the tastant interacts, an ion channel opens, and positively charged cations can enter the cell, depolarize it, and that leads to the activation of voltage-gated channels that ultimately allow for the entrance of calcium at the basal region of the cell, and in a calcium-dependent manner, there's the exocytosis of vesicles that contain the neurotransmitter serotonin. There may be other neurotransmitters involved, but this is one that we know about. And serotonin will interact with receptors expressed at the terminal of that afferent fiber. With sufficient release of neurotransmitter, there will be a depolarization that is sufficient to generate an action potential in that afferent fiber and then communicate the signal on into the central nervous system. Well, other categories of tastants, specifically those tastants that we perceive as being sweet, being bitter, and again this delicious amino acid taste that seems to be distinct in various ways. These tastants interact with G protein coupled receptors. Now, we've talked quite a bit about this category of receptors, so I hope it's now familiar to you. It's a 7-transmembrane protein that is associated on its cytoplasmic surface with G proteins. And these G proteins go on to activate second messenger systems. And these second messenger systems can interact with a variety of other systems within the cell, some of which will lead to the depolarization of the taste cell. Others will release calcium from intercellular stores. So whether calcium is coming in through voltage-gated ion channels or being released from vesicular compartments, the increase in calcium in the basal region of the taste cell is potentially sufficient to lead to the exocytosis of neurotransmitter. So in this particular taste cell, we're illustrating the presence of multiple receptors, and that may be the case. But for many taste cells, there just may be one receptor that is expressed by that taste cell. And this figure gives us more detail than we need to know, but it gives us a closer look at the transduction machinery associated with the receptor. So again, the salts and the acids, they interact with receptors that are on ion channels. And as these receptors are gated, then sodium ions or protons can pass into the taste cell and lead to its depolarization. Other tastants, such as the sweets, the amino acids, the umami tastants, and bitter, interact with G protein coupled receptors. And we know a little bit more about the detail of these G protein coupled receptors. Some of them are monomeric, that is, there's one receptor that interacts with G protein. Some are dimeric, and even heteromeric, meaning that there are different types of receptors. In this case, the TR2, the TR3, or the TR1 and the TR3 that come together to form the functional receptor that interacts with the tastant. In some of these receptor systems, the downstream effector is a TRP channel, specifically a TRPM5 channel that allows for the influx of calcium into the postsynaptic cell, being gated by a second messenger system. In the case of receptors for amino acids and for bitter, we think that second messenger is an IP3 message. So you can see that in various means there are a diversity of mechanisms that allow for the depolarization of the taste cell and the release in a calcium-dependent manner of the neurotransmitter at the basal end of the taste cell.